Hello, welcome to 13 Universal Learning Beyond Barriers. So we're going to be looking at A-level CI biology, all topics paper 4. This is paper 4 series 1. So this is series 1. We'll look at the first question. Remember, this is our first question. This is the first series in A2. This is A2 syllabus. Okay, starting from we're starting from photosynthesis. So chloroplasts carry out photosynthesis. You can see that. Figure 1.1 shows some structural features of a chloroplast and some processes that occur within it. Okay, so that is it. This is a feature of a chloroplast. Chloroplast is a site for photosynthesis, guys. Now bring your writing material. Let's solve this question. It says identify the structures labeled A in figure 1.1. So which structure is labeled A? That's the first question. Where is A? Okay, that's A. Which structure is labeled A? So A is, of course, component makeup. That's A. Makeup of granium. Granium is a stack of thylakoid, and A is a makeup of granium. So A is thylakoid. So A is thylakoid. Okay, thylakoid membrane, thylakoid. So that's thylakoid, all right? So thylakoids are stacked to form granium. A lot of granium forms granule. Let's go to the next question. That's one mark. The same question there. All right, let's go to the next question. Same chloroplast carried out. Okay, so it says explain how the structure and appearance of granium and the components labeled A relate to their function. Explain how. Okay, explain how. Anytime they use the word explain, give us reason, but now they're brought in how. How means that you're going to show a sequence of sequence of events. Okay, so explain how the structure and appearance of granium and components labeled A relate to their function. How? This is very, very important, guys. All right, so the first thing you mentioned is that, of course, they said granium and the component labeled A. This is granium, the stack of thylakoid. Meanwhile, pause this video and try it. This is granium. So how is it? How does the structure relate to the function? Okay, don't forget that granium consists of thylakoid. All right, so each one, this is a thylakoid, is a stack of thylakoid, right? So thylakoid, there are many thylakoid here. So this granium provides large surface area. So we're describing. So large surface area for what? Or why does it provide large surface area? Of course, for light absorption. Okay. All right. Don't forget also that this granium, which consists of thylakoid, the thylakoid, if you look at thylakoid, guys, look at thylakoid. Now thylakoid is actually folded membrane. Of course, it's there, but I just want to show you, right? Yeah. So this membrane here, the thylakoid membrane is this, right? This thylakoid membrane is embedded with photosynthetic pigments or photosystems, right? Photosynthetic pigments arranged in light harvesting clusters called photosystems. So I'm talking about how they relate to their function. So the photosystem consists of light, of course, it's a light harvesting cluster. I call it LHC, light harvesting cluster. So it consists of photosynthetic pigment. What do? They help in the absorption of light, different wavelength of light. Okay, so and of course we have photosynthetic pigment divided into primary pigment and accessory pigment. So the accessory pigment, what accessory pigment does is that they absorb wavelength that primary does not absorb and pass it to primary pigment, and then primary pigment is a site for photo excitation, right? So look at this, guys. If you remember this, just trying to explain it. Okay, I'm just trying to explain it. All right, now This is light harvesting clusters photosystem. So the antenna complex, which are these are antenna complex, right? The antenna complex, they are the accessory pigment. So they absorb light that this is primary pigment, okay? So they pass it on to this primary pigment in order to allow photo excitation and photo emission of surface electron to occur, and that leads to photophosphorylation. Okay, so photo excitation, photo emission of surface electron, and of course the electrons that are emitted enter into electron transport chain. So we have also electron carriers embedded here for what? For electron transport chain. Okay, and then the electron transport chain gives rise to pumping, all right, of protons into the thylakoid lumen, right? Okay, so you see that another feature is thylakoid. This is thylakoid lumen or space. It helps in the accumulation of protons. So protons accumulate here. Okay, they accumulate here. I'm going to spend time to explain this in another topic, in another question. All right. Okay, so this space allows accumulation of protons so that the protons can actually move out from the lumen. They can diffuse out from the lumen, they move out. Okay, from the lumen they move out. All right, and then chemiosmosis occurs. They move out through ATP synthase, which combines which combines ATP and ADP to form what we call ATP. Okay, so all of these help in the function of grana, right? So ATP combines with inorganic phosphate. So let's not waste much time. All right, so that gives us ATP. Is that okay? All right, so let's not waste time. That's ATP here. So we have mentioned a whole lot. So it says component labeled relate to their function. The first function is that this provides large surface area. Okay, that's thylakoid stacked together in granule for absorption of light, and it contains in the membrane here, we have embedded in the membrane here all those photosynthetic pigments which are arranged to form photosystems. They have in absorption, photo excitation, and photo of course photo emission of electron. That's photophosphorylation. Okay, and that helps to move protons. Of course they contain electron carriers which helps in electron transport chain, which also pump proton here. This also consists of this thylakoid space to accumulate protons. All right, let's see what the mass scheme says. Now see this. So anything you mention, you have to state the function. Like we said, stacked many thylakoid. All right, stacked like we said. What's the function? That increase. Okay, that it provides large surface area. Why? To increase light absorption. So the next one is thylakoid membrane has named pigment. They have photosystems, electron carriers. Now these pigments, okay, help in absorption too. But what's the function? Thylakoid membrane is a site for light dependent reaction, okay, which is photophosphorylation. And of course, like I told you, they create electron proton, and of course that helps in chemiosmosis. Chemiosmosis is when we make ATP by joining ADP and inorganic phosphates. When electron is pumped, the electron moves from one carrier to another, energy release is used to pump proton. Okay, and then the proton is pumped into the thylakoid lumen, and when it diffuses out through ATP synthase, that's what is called chemiosmosis. So proton is moved down concentration gradient, and then it is used. The movement of proton causes ATP synthase to join ADP and inorganic phosphate to form ATP. Is that okay? So that's what happens, guys. All right, so look at the next one. Thylakoid lumen, accumulation of hydrogen ion. Like I said, it's a set of proton gradient. Okay, chlorophyll absorbs, right, all of that. So look at chloroplast carrier. Okay, look at the sentence here. They now said identify the metabolic pathway labeled cycle C. Okay, this is Calvin cycle. Okay, it's Calvin cycle, guys. Next question. The same question, but the sub question. Explain the pathway. Okay, why the pathway C is described as a cycle? Can you see it? Why is it a cycle? Now, from what you can see, there's no start or end product molecules. Can see there's no start or end product. You can see that. Number two is that all the molecules are always present. The intermediate molecules are always present. That's why it's called a cycle. Remember that, if you remember the first reaction of this, that CO2 combines, if you remember it, how CO2 combines with CO2 combines with RUBP. Okay, so this RUBP combining with CO2, RUBP, ribose bisphosphate combines with CO2. Okay, and that forms citrate. Remember? Okay, let me call citrate Ci. All right, citrate gives us 2GP. These 2GP molecules, which are 3-carbon compound. All right, citrate is a 6-carbon compound. This RBP is a 5-carbon compound, right? So you can see all these intermediate molecules. This gives us 2TP. Okay, triose phosphate. Okay, 2TP, and then the 2TP, all right, is actually regenerated. It's used to regenerate RBP. So you can see this is a cycle. All right, all these intermediate are always present. RBP is regenerated. That's why it's a cycle. All right. Okay, this first part is called carbon fixation. This react to this. The second part from here to here is reduction. ATP and NADPH is involved here. From here to here is called regeneration. Okay, so why is it called a cycle? Let's look at it. No start and end point. Okay, no start and end point. You can see that all molecules or intermediates present. Okay, ribose bisphosphate is recycled, like I've said. All right, so that is why. Let's go to the next one. The same question. Okay, identify the products of photosynthesis labeled B and D. This is B. Okay, this is the site for light dependent stage. Okay, so this product is going out of the chloroplast, which is oxygen. Right, there are 3 products from light dependent stage. We have oxygen, NADPH, and ATP. So ATP and NADPH, they are needed in Calvin cycle, where the power ATP releases activation energy, provides energy for Calvin cycle. NADPH donates hydrogen. So, but oxygen leaves. This is oxygen. Now, this is a byproduct of this reaction, which is glucose or sucrose or any other sugar, amino acid. Okay. All right. That's it. So the answer here, you can see B is oxygen, D is sugar or hexose, glucose, triose phosphate, carbohydrate, starch. You can see that. All right. So let's go to the next question, guys. So chloroplasts carry out photosynthesis. The same question. Figure 1.1 shows some structural, of course we have seen that. Suggest and explain the importance of glucose and product labeled B. Yeah, so pause the video and attempt it. Okay, just pause the video and attempt it, guys. Pause it. So pause the video and okay, so what is product B, guys? Product B here is what? Of course, we have mentioned is oxygen. So what is oxygen used? Because it says suggest. The question says suggest and explain. So suggest and explain, the importance of glucose and product label. Okay, let's start with B. B is oxygen, right? Oxygen is involved in aerobic respiration, right? To release energy, make ATP, right? It's involved in oxidative phosphorylation. You know that it is the final electron acceptor, okay, in oxidative phosphorylation. Okay, so look at glucose. Glucose, of course, is the source of energy, is a store or source of chemical energy that is released, all right, for growth, all right, because for other cellular reactions. So let's look at what the mass scheme says. So oxygen was connected to aerobic respiration of oxidative phosphorylation. Glucose stated as a source or store of chemical energy, ATP. Energy flow through food chain. Okay, yeah, that's another thing you can add there. So energy can flow through food chain. Is that okay? Let's go to the next question. Number 2, biodiversity can be assessed at 3 different levels. Guys, remember that we are also doing revision. Okay, this video is revision, right, for what we have done in A2. So as you watch it, you try to revise, apart from applying the concepts. So biodiversity can be assessed at 3 different levels. One of these is genetic variation. So they're giving us one of the levels for assessing biodiversity: genetic variation within each species. So each species have genetic variation. Genetic variation is just different alleles, all right? Wide range of different alleles. They have different alleles, okay? So that's genetic variation. Okay, let's go on. The next point says outline 2 other levels at which biodiversity can be measured. 2 other levels at which it can be measured. So pause this video and try it first, okay? This is do it by yourself. Yeah, what are other two other levels at which biodiversity can be measured? All right, so you know that biodiversity has to do with wide range of different habitats, okay, or niche. That's number 1. Wide range, a number of different habitats, okay. So if you have more habitats in an area, the more habitats, the more biodiversity the place has. And the next one is the number of species. All right, number of species, and of course their relative abundance. All right, so number of species, this number orange of different ecosystem habitat, number of species, and their relative abundance. So all right, so look at the next question here. It said to calculate the genetic variation that exists within a species, scientists obtain DNA sequence from many individuals of one species, then count the number of nucleotides that differ when the sequence of 2 individuals are compared. Let's repeat this with different pairs of individuals. They're trying to calculate, all right, different species, of course. Get the DNA, sequence it, count the different, all right, the number of nucleotides that differ, and then this allows scientists to calculate the mean number of differences at nucleotide position along the sequence. Okay, please, the secret to solving paper 4 is must understand the background information very well. Okay, all right, so read this question again. Trying to calculate it. Let's go. The same question. Okay, then the next one says biodiversity can be assessed that we have done that. Explain why scientists use databases and computers to calculate the mean number of nucleotide differences per site. Now, why do you use database and computers? This is bioinformatics, guy. This is asking you for the importance of bioinformatics. Okay, now this database, pause the video and try it. What is the role of bioinformatics? That's what I'm asking you. Try to retrieve this information. Try to do it by yourself. Okay, tell me, what is the role? Okay, now don't forget that the role of bioinformatics, of course, in this question is that it helps to store. You can store information. Okay, store information like store these different nucleotide base sequences for each organism. All right, store amino acid sequence, that's the primary structure. All right, so apart from storage, it also helps us to comparison. So it helps in storage, comparison of biological information. It helps also in, apart from comparison, to process the information, analyze the information. It's very important. And of course, it's also very fast. Let's look at what the mass scheme says. Okay, please memorize this mass scheme in case you see it next time. To store, you don't want to use the word store, contain a multiple or many genome nucleotide base sequence. So it's storing many. To align or compare or search sequences. To process, analyze, calculate large quantities of data. It helps us to do the calculation or process it. You can also share it. Okay, access information from others. I can access information from any, but of course, database through computer, and I use it for research, and also it saves time or it's faster. Okay, guys, look at next question. Table 2.1 shows the mean number of nucleotide differences per site. That's per site of some species. Let's check it out. Look at this. See this species. This is the mean number of nucleotide differences per site. I want to ask you, which one is more diverse? Which one is more diverse? Remember that this is mean number of nucleotide differences. The more differences, the more diverse. Which one has the highest differences here? Okay. Answer that. But if I want to say that, state the genus name of the species that show the most genetic variation. The one that shows this is the most the one that shows the most genetic variation. Okay. Yeah, they say we should state the genus name. All right. This is Anopheles. You see, this is genus species name. Genus species name. The first one is genus generic name. That's genus. The second one is species. So Anopheles is the genus name because it has the highest, all right, variation. Okay, it has a nice number here. So yeah, Anopheles. The next question says state. Okay, table 2 shows the mean number of nuclei. I think we've done it. Yeah. So repeating the same thing. State how many kingdoms of organizations are represented in table 2.1, guys. How many kingdoms are represented? This Drosophila melanogaster is an insect, fruit fly. Fly is an insect. That's Animalia. Animalia is one. Anopheles gambia, mosquito, vector. So mosquito is also an Animalia. That's still one kingdom. They belong to the same kingdom. Plasmodium falciparum, proctotis. Okay. And then sea maize is plantae. Proctotist, Animalia, and plantae. That's three kingdoms. Okay. Three. All right. Genetic variation is considered important in the conservation of species. Low genetic variation is assumed to decrease the chance of long-term survival. Okay, so that means that low species, all right, low variation. The lower the variation, all right, the less the chances organisms survive. Let's look at the next question. So give reasons why low genetic variation may decrease the long-term survival of species. Why? Now I want you to pause this video and then list as many points as you. Okay, I pause it, no add marks here because I just want you to express yourself. We're solving questions, all right? Express yourself. What do you know, guys? Give reasons why low genetic variation may decrease long-term survival. Now, when you say low genetic variation, probably there are fewer. The gene pool is small. Okay, so it has a small gene pool. Do you get? All right, and of course, um, if there is low genetic variation, you guys, you know that there's low genetic variation, that natural selection, all right, will have less variation to work on. Natural selection, the raw material for natural selection is variation. So when there's a change in environmental factor, all right, it will affect most of the organisms. Okay, because the variation, they have little variation. So what affects one may affect the others. So change in selection pressure affects almost all the organisms. Okay, and so we have less organisms able to adapt, or survive or evolve. Which you get. So change in environmental conditions can affect them, and then of course small gene pool, of course fewer organisms available. All right, that is it. Let's go what the mass scheme says. Okay, look at it. 5. Less able, not able to adapt. So few. Okay, the organism cannot adapt in that environment. There are very few. Little variation for selection to act on. Variation act on, um, of course, is the raw material for natural selection. Fewer potentially useful. Yeah, so there will be fewer or beneficial alleles. Remember that organisms that have beneficial alleles, they are able to adapt and survive. All right. So if we have small genetic variation, that means we're going to have less less alleles. All right, less advantageous or useful allele. That's what it's trying to say. Okay. So almost organisms could be killed by the same disease or selection pressure. Now, if due to small population size, this decreases special. Okay, so small population size will decrease survival. All right, that's 3 marks, just 3 marks, guys. Fig 2.1 shows how the International Union of Conservation of Nature categorizes species according to their conservation status. Now, common species with lowest conservation at risk of extinction are categorized as least concern. So you can see this is extinct. This one is going to say extinct in the wild. This is extinct everywhere, both in captivity and the wild. Okay, critically endangered, okay, that's increasing. Okay, risk. All right, so you can see that as we go up. So let's look at what the question says. It says, question 2D states that low genetic variation is assumed to decrease the chances of long-term survival of a species. We said it, right? Because there'll be fewer beneficial alleles, and then less organisms are able to adapt, small population, all right? And of course, change in selection pressure will affect all of them. So that's what it's trying to say here. It's a predict the relationship between genetic variation and conservation status. If this assumption is true, if the assumption is true, okay, so you see that the less the genetic variation, okay, the higher the status. See, this is increasing. So that means that the ones that have less genetic variation are less likely to go into extinct. Okay, so the ones at the base are likely the ones that have um the more genetic, all right, variation. So the more genetic variation we have, the lower the status on conservation. Okay, the lower the conservation status. So these ones have more. That's what I'm trying to say. That these ones have more variation. Okay, because they are the base of this conservation status. The one at the top here has less variation. That's why they can easily go into extinction. Okay, that's what they said. The answer says lower genetic variation means higher conservation. Yes, this lower, yeah, that's higher conservation status in vulnerable intentional capacity. Okay, so this guy has of course higher conservation status. He has lower genetic variation. Okay, so guys, let's go to the next one. Fig 2.2 shows the mean number of nucleotide differences per site of some species and some species of mammal and their conservation status. All right, that's it. This is mean number of nucleotide, okay, differences per site. That's the organism. This is the conservation status. You can see that. All right, this assess whether the data in Fig 2.1 provide support for the prediction you made in 2D. 2D says the lower the genetic variation, all right, differences, okay, the higher the conservation status, the more threatened, all right. So does this support it? Does it support it? All right, pause this video and try to solve it. Pause this video and try to solve it, guys. Now if you check, of course, you see, so the one, if you look at here, because this is mean number of differences, so the one that has the highest number of differences is chimpanzee. So where is this conservation status? That's endangered. So this doesn't support it because the mean number of nucleotide differences here is high, and then it's at a higher, part of the conservation status is endangered. It does not support it because chimpanzee has higher mean number of nucleotide difference, so it should not be endangered. Okay, this does not support it. And if you let's check the one that has the lowest. All right, okay, let's check this one, the one that has, okay, common minke whale. Okay, this is common minke whale. All right, you see it has the lower status. Okay, so least concern. Guys, can you see this is testing your analytical skill to spot out trends and to evaluate, okay, data. This one, based on that hypothesis, should be critically endangered. Okay, but in this hierarchy here is at it has the status is least concern. Okay, so it does not support the prediction that the smaller the differences in their, okay, nucleotide differences, the higher their conservation status. Okay, if you check on that one, see lion is something vulnerable. Okay, so I've mentioned it. So let's look at what the man scheme says here. It says any three does not support because chimpanzee has most or higher genetic variation. That's it, but it's endangered. Number two, minke whale has lowest genetic variation and least concern. Three, genetic variation similar or same but conservation status is different. Okay, yeah, genetic variation is similar. This is similar. They should have the same. A lion and common mink are similar. All right, they they should have similar conservation status, but one is least consigned, which is common mink whale, and one is vulnerable. So it doesn't support it. To give examples, these are things you mentioned. Gorilla is most critically endangered. See it. Most critically endangered is gorilla, but it has high genetic variation, so it doesn't support it. All right. So you have to look at the data and then make a decision. All right. There are more than 600 plant species in the genus Ipomia. Many species are grown for their attractive flower, and some species are used as crown plants. Fig 3.1 shows Ipomia purpurea, the common morning glory. You can see that. All right, look at this question. It says the gene that determines flower color in Ipom, this is purpurea, not purpose, has two alleles. A dominant allele that results in purple color. So the dominant allele results in purple color. Recessive allele that results in red flower. Okay. A student recorded the flower color of all the arboreal plants in a field. The students recorded 660 plants with purple flower and 440 plants with red flowers. Okay. Assuming they had a one-batch principles applies to the population, calculate the number of plants in the field that are heterozygous. Calculate it. So these are the equation P plus Q equals. So this is 1. This is also 1. So pause the video and then try to solve it. Okay, show your workings, guys. Let's go to work. Remember that this P here represents frequency of dominant allele, Q represents frequency of recessive allele, but P squared represents frequency of individuals that have homozygous dominant phenotype. Okay, or genotype, yeah, the phenotype. Okay, this equation represents the frequency of phenotype and genotype, right? Why this is just allele. So let's get to allele and use it to solve for the individuals that are heterozygous, which is 2pq. P squared is individuals that are homozygous. This is heterozygous, but two of them are dominant, right? So what we're given, we're always sure of the recessive allele. Remember that they told us the recessive allele. Look at the recessive allele here. The recessive allele that results in red flower, which is 440. Okay, there's only one recessive. Okay, this is it. All right, this can be shown as this and this. So let's look at it, guys. So half for recessive allele, let's get it for the which is Q. Right, so Q squared, solve it, get your calculator, is equal to, of course we are solving probability, which is total number of recessive plants, which is 440. All right, all over the total number. Total number is what? Is 1. Okay, add it. Let's see. That's 1,100. 1,100. Okay. By the time you divide this, it's going to give us 0.4. Did you get 0.4? So if you want to get solve for Q, therefore Q should be what? Square root of 0.4. So I want to calculate for Q. Square root of 0.4. Square root of 0.4. Okay. All right. So if you find the square root, it's going to give us, what do we have? 0.632. 632. That is it. 0.0632. Now, of course, we have gotten Q. So P will be what? Remember that P here is P plus Q equals to 1. This is 1, please. Let's type 1. Okay. So that means that P will be equal to 1 minus Okay, 0 point what? Yeah, 0.32. Good. So we have used Q to find P because Q is recessive. Recessive is sure, right? But dominant could be heterozygous, homozygous. Do you get? That's why we use recessive to find the dominant. Okay. So yeah. So P is what? 1 minus that will give you what? Is somebody punching their calculator for us? That will be 0.36368. Okay, that is it. 0.368. 0.368. Yeah, that's P. So remember that they said we should find the ones that are heterozygous. For what? They said we should calculate the number of heterozygous plants. Is only one that's heterozygous. That's heterozygous dominant. Okay, 2pq. So 2pq is what we're looking for. Now we know q, right? We know p. So we just multiply them. The frequency times the total number. The frequency is 2 times p. P is what? 0.368. 0.368. So help me punch the calculator. Okay, that's 8 times what? Q is 0 point, right? 0.632. And then times the total number of all the plants, which is 1,100 plants. Okay, 1,100 plants. So what do we have here? So we're going to have the answer will be what? Just multiply for me. Okay, just help me multiply it. Okay, let me do. I have a calculator here, so I'll just say 2 times 0.368 times 0.632, and that gives me 2 times 0.368 times 0.632. Okay, all right, times 1,100. Okay, all right. So what's the answer there? Let me try it again. I think there's a mistake. 2 times 0.368 times 0.632 times 100 and 1,100. Okay, yeah, that's 511. Yeah, we're correct. 511. Okay, that's heterozygous. Okay, point something. All right. Let's see how they did it. Okay. So you see how they started? Yeah. So you have to start Q. Q represent Q squared represent the frequency for recessive allele. Okay, recessive phenotype, right? So because it's always one, dominant can be heterozygous or homozygous. So the same thing we did. The answer. The Japanese morning glory eye has over 20 different flower colors phenotype, including the shades of blue, purple, red, and pink. The flower color of eye nail is controlled by at least 4 genes. The flower color can change gradually after the flower open each morning and can change with fluctuations in the carbon dioxide concentration of the surrounding air. It is then concluded that the flower color phenotype in eye nail shows continuous variation. So there's 2 reasons why students made this conclusion. Now, continuous variation. Pause the video and try it. It's controlled by more than 1 gene. This is 4 genes. Controlled by more than 1 gene. That's number 1. Number 2 is affected by environment. Okay? They said it can change with fluctuations in the carbon dioxide concentration of the surrounding air. So it's affected by environment. It has intermediate. Are you following? How do I know it has intermediate? Okay? So of course we have different colors. We don't have distinct categories. Okay? So don't waste time on that. So color shades, categories, no discrete overlap. Okay, more than one. Okay, further by environment. All right, look at the next one. Scientists investigated the response of stomata to changing carbon dioxide concentration. That's carbon dioxide in the beach. Okay, morning glory. The scientists place Ipes caprea plant in chambers. They measured the width of open stomata aperture. After the plant had been exposed to different CO2 concentrations for 40 minutes, the light intensity and temperature were kept constant. The relationship between CO2 concentration and the mean width of stomata aperture is shown in figure 3.2. So that relationship, as carbon dioxide concentration increases, what has happened to mean width of stomata aperture? It decreases. So that's negative trend correlation, right? The question says in 2016, our study measured the atmospheric CO2 concentration as this 400 micromole per mole. In the future, climate change may reduce water availability and increase atmospheric CO2 concentration in some habitats. Suggest how the stomata response shown in Fig 3.2 would allow this organism, Ipes capre, to survive the effects. Okay, so remember they said it in future. All right, the climate change may reduce water availability. So that means there's going to be a problem. That's less water availability. Will condition will be created, right? Conditions with less water availability. That's like desertification. All right, and you know that in desert environment, more water is lost. Okay, and most water that is lost through stomata. So how is this plant going to survive? State suggest how the stomata response. There's a problem. How is it going to survive? How stomata response shown would allow this to survive the effect of climate change. How it survives is, look at it. You know, as CO2 concentration increases, the stomata aperture will decrease. So in such environment, the stomata aperture will close, or it will decrease. The aperture will decrease as CO2 concentration increases, that causing less water vapor loss due to transpiration. That's it. Hmm? So increase causing stomata aperture to decrease, stomata will be less open, less water loss operated. So the plant survives. Okay. Under certain condition, the closure of stomata is controlled by abscisic acid. Describe how abscisic acid causes the closure of stomata. Guys, remember the first thing that happens, abscisic acid binds to the receptor on the membrane of gut cells, and then hydrogen ion no longer pumped out. Okay, so if it's no longer pumped out, calcium ions channels open, and the calcium ions moving. When they move in, calcium ions act as second messenger, right? It causes of course potassium ion channel to open. Of course, before then, the channel for negative ions, chloride ions, will open. Negative ions will move out. When they move out, then it creates electrochemical gradient. So we have more positive ions inside the gut cells. So potassium ion channels will open, potassium ions will move out. Once they move out, water potential of the gut cell increases, and then water leaves by osmosis, and then it loses sugar. So look at it. Binds receptor on gas cell surface. Hydrogen ion stops moving out of this. It stops moving. Calcium ions enter. Calcium ions act as second messenger. Potassium ions leave cells, and water leaves by osmosis. Okay. All right. Down water potential. So that is it, guys. Just 4 marks. Make sure you memorize this and remember it. This is how it works. All right, guys. Look, at scientists are researching whether abscisic acid can be used in crop treatment to increase yield. Evidence suggests that abscisic acid modifies the effect of auxin on elongation growth in plants. So scientists investigated the effect of different concentrations of abscisic acid on root elongation in seedlings of Telecress Arridopiosis thaliana. The seedlings were divided into 4 groups. You can see that. Control is 0.0. 3 experimental groups, each treated with different concentration of abscisic acid. See the different concentration for each group. Seedling root was measured for 6 days during the treatment. You can see that the rate of root elongation was calculated each day. The results are shown. Look at the result. Okay, so the results are shown. Yeah, guys, So please, anytime you're dealing with graphs, please make sure you read the keys very well and check what is on the x-axis. That's independent, this is dependent. So as the day increases, what's happened? Okay, in three of them, rate of root elongation increases, except for this one. Okay, this is the highest concentration of abscisic acid. All right, yeah, you can see that the one that has control and the one that has 0.1 mole for abscisic acid, the one that has 0.1 mole for abscisic acid, that's it, the one that one increases more, all right, than control and 1.0 micromole of abscisic acid. So that's what they want you to do to describe this graph. What does it mean to describe it? Show the general trend first. Okay, in these three, okay, the rate of elongation increases with time. Okay, but this one remains constant. That's 10 micromole of abscisic acid remains constant for 3 days, and then it increases. Okay, and then of course stays constant and then decreases. But if you come to, let's look at the specifics here is that the one for 0.1 mole of abscisic acid increases more than others, more than that of 0.1 mole, which is this, and control. So control and 1.0 mole, these two, they are higher than 10 mole of acetic acid. That's what I wanted to say. So we'll describe the effect. That's it. That's the effect here, guys. 0.1 mole increases gives higher rates compared to control. Just compare them, the general trend, and you compare them. This and that decrease give lower rates compared to control. Yes. So this stays approximately constant. You quote data to support it. That is it, guys. Look at this passage outlines of the role of oxygen in elongation growth in plants. Complete the passage by using the most appropriate scientific term. The binding of oxygen to receptors causes, this should be hydrogen ion, to be pumped out into cell wall. This activates protein called expansins, which disrupt the link between cellulose microfibrils. The cell walls are then able to expand. That's it. So protein and cellulose fibrils. Okay. Sorry for taking your time, guys. This is where we'll stop for now. Thank you for watching. We want you to share our videos, make comments. All right. So see you in series 2. Bye.